That sounds good. Yeah, no, I'm happy to take questions as we go. If we don't finish, I'm fine with that. I decided to talk about our metabolism studies. So this is a little bit of a tour of the Rathmell lab over the 20 years that it's been around. I have conflict of interest stuff, which is almost entirely my husband, who is dabbling in starting companies and stuff. So I'm going to talk about the metabolic demands in tumors broadly. We'll start with the tumor cells and then work our way out from there. So I actually start this talk with a couple of cases. These could easily have been patients that I saw, but they handily, someone else saw them and reported them. And so you can look them up if you want. The first one, a 22-year-old man with hypertension and mass on the top of his right kidney, has a PET scan, and the PET scan lights that mass up, but he also gets a CT along with it, and the CT identifies that there are multiple kidney lesions in addition to what looks like a pheochromocytoma. Interesting case. You probably thought we'd be talking about 65-year-old men, but instead it's a 22-year-old with a non-PET avid kidney tumor, but bunches of them, and you can see them here in the. Actually, does that arrow show for you? Okay. My screen is doing like, it's on the wrong screen, so I apologize. I keep looking over to the right. Okay, I have another case. So similarly, another young person, 24 years old, who presents with abdominal discomfort, anorexia and weight loss, and has a massive tumor. It's arising from the kidney. It's a cystic mass that's intensely PET avid, and even has some metabolic deposits. I didn't show the pictures. They're also intensely PET avid. So we have two young people with tumors arising from the kidney, one very PET avid and one not. And let's go. So before we go to what those two were, I think about kidney cancer and trying to remember that these tumors arise in the kidney, which is an interesting place for things to happen, right? It's not just any cell. These are cells that live in a complex environment that has the most extreme gradients of glucose, oxygen, salts, and toxins of any part of the body. So most of the tumors that we study come from the cortex and really from the proximal tubule, which is the most glucose replete, the most oxygenated place in the body. So that's where it all starts. And then I'm going to give you just a little bit of history lesson. Both of the patients that I presented will turn out to be familial cases of kidney cancer. And if you have an interest in familial kidney cancer, it's important to know that there's a really nice history to this. Bert Zabar was at the NCI. He's a geneticist who saw that there were these clusters of families, this is in the 70s, of patients having early onset kidney cancers. And of course, this was von Hippel-Lindau disease, and there are a few others. And so he actually went out, he was called the Yellow Beret geneticist. He went out and studied these families, literally went to their homes, asked them all to get together, tell their family history story, and then collect blood. And then did old school genetics to clone the genes that led to us understanding these diseases on a different level. He worked really closely with a urologist who's still very active in kidney cancer at the NCI. So when I was joking earlier, Marston was always a kidney cancer person way before me and Brian. But together they cracked the genetic underpinnings of a variety of these hereditary kidney syndromes. So we'll talk for a minute about VHL, the von Hippel-Lindau syndrome, and you guys know the genetics of this very well, but also found the MET oncogene papillary renal tumors, fumarate hydratase as another cause of papillary type 2 renal cell carcinomas, and also identified the Berthog-Dubé gene leading to chromophobe and oncocytoma. So these guys single-handedly identified all of these diseases. So let's go back to the kidney. So if you have cells in the proximal tubule, that we have at this time no way to know that there's any stem cell for cancer here, or if there are subsets of cells that are more likely to become a clear cell type with a VHL mutation or a papillary type with an FH mutation. It looks like they all come from the same lineage. clearly a Paxate lineage cell. So, but here you've got, if you lose VHL, you get upregulation of HIF in this very angiogenesis dominant disease, very, a lot of blood supply enriched to these tumors. But if you lose FH, so that's, you know, down there in the Krebs cycle, you get a much more metabolically dominant disease, and they're very glucose avid. So that's some clues to what our patients had. So patient 1 had a PET negative kidney tumor, and as well as pheochromocytoma. So that's the clue. That's a von Hippel-Lindau patient. Patient 2 was intensely FDG avid and is an FH mutant patient. So these are these syndromes. So that's actually all I see anymore. I don't see anybody with regular old metastatic kidney cancer. I just see these hereditary patients. So VHL disease is carrying a germline mutation in VHL, and then carries risk for, in addition to clear cell renal cell carcinoma, cerebellar hemangioblastomas, spine and retinal hemangioblastomas, and pheochromocytomas, and pancreatic neuroendocrine tumors. And HLRCC, the hereditary leiomyomatosis and renal cell carcinoma, comes from deficiency of the fumarate hydratase gene. And they have also early onset kidney tumors that are these very aggressive papillary type 2s. They also have some interesting fibrofolliculomas in the skin as well. And they get uterine myelomas, leiomyomas, and they can have other leiomyomas as well. But they're, the two diseases are very contrasting. Although they both occur in young people, the VHL-driven clear cell renal cell carcinomas, histologically different and very slow growing typically. We actually watch them for years before we take any action. And the fumarate hydratase tumors, when they come, they just explode. So they're very, very aggressive tumors and very hard to manage. But they share some common links, in particular HIF. So the common link is just regulated oxygen sensing, and the Nobel Prize, this is Bill Kalin, who was studying this connection between hypoxia signaling and kidney cancer development. And so that's the subject for this Nobel Prize. And so HIF is upregulated in both of these tumors, but is more clearly a driver, the HIF1 in the FH, and HIF2 maybe in the clear cell type. So we're mostly going to talk about VHL-driven renal cell carcinoma because that's a better model for most renal cell carcinomas. FH, I think, is a little bit fringe, but it's super interesting biology, so it's good for comparisons. So VHL-driven renal cell carcinoma, here's how I think about it. Here's your normal kidney cell, and minding its own business with VHL taking away HIF out of the system in an oxygen-dependent manner. But you lose VHL, and now HIF is free to accumulate. It's a good potent transcription factor, turns on all these genes, among which lots of them are transcription factors that are transcriptionally regulated metabolic factors. So this influences the ability to take up glucose and shunt it through to lactate, like an aerobic glycolysis, but also promotes the uptake of glutamine and reductive carboxylation.
Kim, can I ask a question?
Yeah, for sure.
So this is the only published article in which a connection was drawn to SLC1A1 and 1A3 that I have found between VHL loss and SLC1A1 and 1A3 upregulation. We have been interested in this for a completely, we identified SLC1A1 as a candidate oncogene from a genetic screen. And so we are hoping that we'll submit this story pretty soon.
Hey, Abhishek, you froze. You froze for
really?
there. Yeah, for about the last 10 seconds.
It's HIF, it's VHL dependent transcriptionally. And we've done this like in multiple different ways that you can think of. So I was wondering, what is the, like, am I missing some evidence in literature that links these metabolic pathways to HIF that I need to reconcile over here? Because for the life of me, I cannot get it to be a HIF target gene at all.
Yeah, I don't know that that the glutamine transporter itself is a HIF target. We've actually seen more that that it affects some of the enzymes that modify glutamate. So like GLUD1, I think those are HIF targets. I'm not sure that this is actually a HIF target.
Okay. Yeah. So because SLQ1A1 and 1A3 actually they take up aspartate and glutamate both from extracellular
they
space.
do. That's right.
And so which we have evidence for, but they don't seem to be responsive to HIF at all. Like we can remove HIF from VHL deficient cells, no impact. We can put back HIF in VHL proficient cells, no impact. We can try the pharmacological regulators, no, for almost certainly not a HIF target gene. But definitely very strongly VHL regulated.
But VH, oh, I see. That's really interesting. Well, I mean, there are other VHL targets, right?
Yeah, so that's what we are chasing down now, transcriptionally, what it might be, what is the key mechanistic transcription factor here. But that's what I just thought I'll ask you, because this was the
that's a really good question.
Only cartoon that I have seen in which those two have been linked together.
Oh, yeah, it wasn't meant to show that it was HIF dependent. It was just meant to show like how does the glutamine get in.
I understand.
Thank you. Okay, cool. But I'll talk to my husband's a transporter guru.
Yeah, I'm happy to share this data with you guys and then see what your thoughts are. Yeah, definitely.
Okay, cool. Just to talk about PET scans for a minute, we'll start with PET scans and then end with PET scans. So PET scans are widely used in cancer, and were another Nobel Prize was the recognition that there's altered glucose uptake in tumors. The experiments themselves were taking slabs of tumors and then put them on like an Erlenmeyer flask and look for the evidence of glucose utilization, and tumors did more than quote normal tissues. This was not done in tumor cells per se, although there are clearly some tumor cells that are very intensely take up glucose. Anyway, I'm getting ahead of myself. So based on this finding that tumors could preferentially use glucose as their energy source, we started doing PET scans, right? So FDG glucose is used for a lot of imaging. Here's a lung cancer patient. I'll remind you though that if you do a PET scan on a patient who just had a SARS-CoV-2 COVID vaccine, they will also light up. So immune cells also use glucose. You can see actually in both of these figures that the brain is intensely lighting up. So there are a lot of different kinds of cells that have a glucose predilection more than other types of nutrients. Okay, so that's called the Warburg effect. Let's talk a little bit more about what that really is. The Warburg effect is when you go from a normal cell, takes in some glucose, converts it to pyruvate. Pyruvate gets shunted, into the mitochondria so that you can make energy. You can make ATP utilizing oxygen. But with Warburg effect, the glucose is taken up in much higher levels, makes a lot of pyruvate, and it is not going to the mitochondria, it just goes to lactate. So this is what we call the Warburg effect. And so I just told you that cancer cells like to take up glucose. We showed that HIF, when it's activated, activates all kinds of things that upregulate metabolism pathways like that brings up glucose. And yet the first slide that I showed you was a VHL patient who had a glucose or a PET cold kidney tumor. And so this is something that has been a conundrum for me for a very, very, very long time, why we can't use PET scans. This should be the tumor that is the poster child of PET scans. And if you've ever done a PET scan on one of these patients, they might light up maybe more in their mets than in their primary, but they really don't. There's really a disconnect here where they are not taking up glucose at the same kind of rate as we might have expected of a tumor that's got the pathway we think we understand. So we spent a lot of time trying to understand why this might be the case. A lot of this glucose avidity has been tied to HIF1. The HIF family is, of course, more than one. There's HIF1 and HIF2, these paired transcription factors, both of which are downstream of VHL. And so one possibility was that HIF1 is giving you this glucose metabolism phenotype, and HIF2 we know drives a little bit more of a de-differentiation and the polycythemia and other nutrient uptake, although GLUT1 brings up glucose anyway. But that maybe this side of this activated pathway was the Warburg, and this side was, I guess I'll go with not Warburg. And that actually made sense for a minute. We showed a long time ago that that tumors came in a few flavors. They were VHL wild type. There's a few of those. Those are clear cell tumors. Or they were VHL mutant and expressed both HIFs, or they were VHL mutant and they only expressed HIF2. We actually never found one that only expressed HIF1. And so we thought that was potentially a way to classify these tumors. And it could be that these HIF2 driven tumors were the ones that were less glucose avid. And so maybe, it was just some numbers game, because there's not a lot of good data on the PET scan avidity in kidney cancer. It's usually a very small case series. And then Bill Kalin's group came out with a really beautiful paper, and I won't go through what all this data is, but if you look at these graphs down here at the bottom, these are all looking at HIF1 and HIF1 sort of genetic copy number presence. Full presence is up here at the bar and absence is gone, right? So the HIF1 gene is being selectively deleted in a lot of these tumors, but not just gone, not the whole gene. It's just like portions of the gene are gone. Actually, so much so that maybe some of these tumors that we'd seen before expressing HIF1 might be expressing a portional HIF1 that was maybe missing one of these small deletions. For example, A498 is a very classic cell line. A lot of you may use A498. We use A498 all the time. We thought that it was a HIF1, HIF2 expressing cell line, but it turns out the HIF1's non-functional. It stains nicely with an antibody, but it doesn't actually do anything. So, so that actually made us feel even better. These were all HIF2 tumors. HIF2 was not the Warburgian HIF, and maybe that was the reason why PET scans weren't any good. We also looked at cells and asked, if we overexpressed one or the other, could that help us understand? And that actually was a little bit confirming for us. This was a study we did now several years ago where we took genetically engineered mice that had a stable HIF1 or HIF2 engineered into their kidney cells. It was kind of overly complicated. Could have done this in a much easier way. But anyway, they overexpressed HIF1 or HIF2. And then we did seahorse assays and said, ECAR measures like glycolysis essentially, and OCAR measures oxygen consumption. So this is extracellular acidification. So this is a measurement of glycolysis. And you can see if we overexpressed HIF1, you do more, right? So they're doing a lot more glycolysis. But when we overexpressed HIF2, actually, if anything, they did just a smidgen less. Not really significant, but a little bit less. And the same thing for oxidative phosphorylation. They did more when we gave them HIF1. So they're doing glycolysis, they're doing oxidative phosphorylation. It was really interesting. The HIF2s didn't want to do glycolysis, but they're still doing the oxidative phosphorylation. And I don't think I actually showed it in here. We showed that these guys really, the HIF1s really liked to take up glucose, but the HIF2s really preferred glutamine. And so then it made sense that these are really doing reductive carboxylation. If somebody could mute
There is a
background.
Thank you.
Yeah, thanks. Okay. So then we started just doing some PET scans on patients. So this was a study where we did PET MRI and then did regional analysis of the tumors. It was really interesting because we took big tumors. We sent those patients in to get their PET MRI before they went to surgery. And then we could, this was literally days before surgery, then we would take that map and go in and identify regions that were PET avid or PET cold. And it was variable across the tumor. Actually, a lot of them were cold, but some of them had small regions of higher uptake of FDG. Here's just one example in this patient's tumor. And so we're able to go back in and take a sample from here, very PET avid, over here, more PET cold, and here somewhere in the middle. And then stain those for features. One, we didn't see a lot of HIF1. That's fairly convincing. That was not well matched to whether they, oh, it cut off at the top, whether they were FDG avid or not. They're most avid on the right and less avid on the left. The things that corresponded were GLUT1, so the glucose transporter. That makes sense. Finally, something that corresponds well. And fructose bisphosphatase 1 is lost in a lot of tumors. Celeste Simon has a whole series of papers now on the role of losing this vital enzyme of glycolysis. And when it was gone, that was when we had the most glucose uptake, which is really interesting. You basically uncouple glycolysis and now suddenly they're taking up more glucose. It almost suggests there may be some feedback that they're not processing it well, but they're choosing to take up more. So we've seen tumors are displaying an altered metabolic property of at the functional and tissue level. That's the PET scan, but down in the cells, they can shift their metabolism depending on genetic features, meaning if it's a HIF1 or HIF2, that tells us one thing or another. And that the classic Warburg effect may require the more high-level HIF1 as opposed to just upregulation of HIF2, or maybe other metabolic uncouplings like the FH mutation. So FH is going to uncouple the Krebs cycle entirely, and so that may make a true dependency on glucose, or it may be, we know when you uncouple that, you actually shoot HIF1 up, and maybe the levels are actually so high that that it allows them to be glucose dependent. Okay, so that's the cancer cells. But in the tumor, there's a whole bunch of other cells as well. So I'm going to shift gears and talk about the immune cells that are present in there. This is all the people who were involved in this study. I happen to be married to an immunometabolism guy. So this is Jeff Rathmell. And in his lab.
Can I ask another question? Sorry.
Yeah.
Following upon Amato Ghiaccia's findings on SLC2A1 as a potential target from his pharmacological screens,
yep.
If you go back and check if the more glucose avid tumors are also the ones that are GLUT1 dependent or independent, is there a correlation there?
Yeah, that's a really good question. We did not look at that to my knowledge. I'm just trying to think if we did in any. Yeah, this is old enough that we weren't doing RNA-seq like every time we turn around, and we couldn't do single cell. That was a decade ago. We did a panel of genes, and I don't think that was on our panel.
I do remember, and this is again very old papers, but I do remember with the isogenic cell lines, 2-deoxyglucose did seem to be synthetic lethal with VHL deficient cells, and made us think that, oh, maybe these are again going to be HIF high, glycolysis high cells. But I'm now wondering whether we should be thinking more in terms of FBP status and HIF1 versus HIF2 status before we make those comments.
Yeah, I do. I do think that those are two features of cancers that will play a role in whether or not they're going to be sensitive to detectable with an FDG PET scan, or I don't know about that data on the sensitivity to 2-deoxyglucose. We've played a little bit with that, and I've found that to be pretty tricky to, and usually at doses that would not be tolerable in humans.
Got it.
Right. So.
Thank you.
But 2-deoxyglucose is a pretty rough inhibitor of glycolysis. So, but GLUT1 inhibitors are out there and finding their way into clinical trials.
Yeah, I think they followed up their original paper. Like there are a couple other stories that they followed up with, right? So I think the postdoc from his lab, she now has her own lab in Canada, I think, and then she has followed up with a couple other stories.
Okay. Yeah. So I'm going to move on. These are the folks in the lab who worked on this. They're all on the papers. One I do want to shout out is Katie Beckerman, because she now has her own research group and works closely with both us and Brian. So another kidney cancer investigator out there. So now I'm going to look at T cells. And this is a bunch of work looking at T cell metabolism in cancer and immunity. There's a little bit of background here that is largely the Jeff Rathmell lab, really exclusively the Jeff Rathmell lab. This is really all they do is study how T cells go from resting to stimulated. When they do that, they shift from oxidative catabolism to an anabolic form of metabolism, and then they shift again when they move into memory state. So what many years of data here show, I'm just going to pull a few more things up, is that the metabolic fuels that a T cell uses are actually very dependent on programs in those cells and are important for the functionality of those T cells. Okay, so just as one example down here, this is IBD, inflammatory bowel disease model in mice. And if you knock, actually this is really important for the questions you were asking about GLUT1. If you knock out GLUT1 in T cells, you really dramatically change the course of the disease in these mice, right? So normally an IBD mouse, you initiate the disease and then they start to lose weight because their IBD kicks in. But if their T cells don't have GLUT1, then they can't activate and they can't get that immune response. And so then the mice are fine and their weight doesn't change. On the other hand, shown up here, if you overexpress GLUT1 in T cells, then you can actually enhance immune responses here, causing autoimmunity. So I think these are kidneys. So this is showing kidney from a transgenic GLUT1 overexpressing in T cells mouse, and they have all these deposits in their kidneys from those immune cells. So you can shift the repertoire of the T cells just by changing GLUT1 and the glucose uptake of those cells. On the other hand, glutamine is also really important. And if you block glutamine, the cells will shift to taking up more glucose. That's what they're showing here. And they actually can become more responsive. So here they're using CB839, which is a glutamine synthase inhibitor. And now these cells are taking up more glucose, which I'm not showing, but they're also activating more. So this is activated T cells. And they become actually dependent on this glycolysis. So this is ECAR, like the seahorse extracellular acidification assay. And so treated with CB839, this glutamine inhibitor, they now are more glycolytic, the TH1s, but the TH17s become less glycolytic. So you actually skew your T cells a little bit. And here we're showing treatment with 2-deoxyglucose. Can have some of that same effect, but if you use both, you actually block those cells from activating altogether. So, so here's vehicle treatment with CB839. They're a little bit more stimulated. 2-deoxyglucose takes them back, but both of them now they don't stimulate at all. Okay, so that's T cell history. So in the tumor microenvironment, we've got cancer cells. In kidney cancer, we've got a VHL mutation, lots of HIF1, and all of these things going on in the cancer cell. And in the T cell, we know that they're very dependent on their nutrient utilization. And so the question was, number one, are these cancer cells influencing the T cells? Are they changing the microenvironment in a way that alters the T cells? And are the T cells that are present in the immune environment even functional? So it's an important question, one, because we're using all of these immunotherapies. That actually might be the next slide I have. But also T cells are really very present in kidney cancers. So this is just a lineup of the big TCGA project looking at CD8 expression. So CD8 positive T cells. And actually the highest proportion of CD8 T cells is in clear cell renal cell carcinoma. And they're not in little nested clumps like in melanoma. They're nicely distributed all around throughout the tumor. So it's actually a nice setup for these T cells to be activated and really impact the tumor. Okay, so, and this is what I already alluded to. Of course, there's lots of immune therapies that are in play. You guys all know this, so I'm gonna skip that slide. Okay. When I'm giving this talk, often I am talking to an audience who doesn't know about kidney cancer or the fact that immunotherapy really can work. And I show this patient case, and this patient may follow with you now, Brian. I don't know what's happened to him. I think he's still doing well. Before immunotherapy, this guy had a big kidney tumor and a bunch of liver mets, and he was on single agent nivolumab. And after a while, his kidney tumor was smaller and we couldn't see the liver mets anymore. And so we sent him to the surgeon because he'd been on treatment for about 2.5 years, resected this, and for at least 3 more years, he was fine. And then I stopped seeing patients with kidney cancer. So I'm not sure where I'm assuming he's still fine. But the question is, why do some patients respond really beautifully like this, right? And others don't, when their genetics, at least of their tumors, seem about the same. So it seems like it may not be about the tumors. It may be more about the immune cells. And I'm going to skip that one, actually. So kind of going back to this heterogeneous immune environment of the tumor, we know that in this environment, there is some hypoxia. We know there might be, or at least it was widely assumed, that there was competition for glucose. You know, there's a finite amount of glucose around, and we think that the tumor cells should be wanting it. We think the T cells should be wanting it. So, you know, does somebody win or lose? There's thought to be a depletion of amino acids, an accumulation of lactic acid from all of that Warburg effect. And some of those might be immune suppressive signals. So the question was whether the microenvironment actually functioned as an immunometabolic checkpoint. So here's back to, this is our cancer cell with all the stuff we think about in the cancer cell. Here's the T cell with all the stuff they think about in the T cell, the glucose driving a Th1 and the glutamine driving a Th17 type phenotype. So we started just going into human tumors and pulling out T cells, and we stuck them on a CyTOF just to take a look at what their phenotype would look like. So this is first looking at some control T cells. This is actually peripheral blood T cells, and you can see here's CD4 T cells, here's CD8s. There's not so much in whatever these other components are. So this is just sort of a high level phenotype grouping of T cells, okay? And if you were to stimulate these cells by treating them with CD3, so now they start to activate and they shift and they have different markers, and so the CD4s pop over into this grouping, the CD8s pop over into this grouping, and okay, so these are our controls, resting and stimulated. So we pull TILs, tumor infiltrating lymphocytes, out of kidney cancer patients, and we stick them on the same assay, and they're not in either place. They don't match up as resting, they don't match up as stimulated, they live in kind of their own in-between space, which is super cool and interesting. This is the features that make up the different spaces. And so the question is, you know, are they alive? Are they dead? Are they stimulated? Can they activate? Can we make them activate a little bit more? And I wish I had this guy animated, but he'd move really slowly. The answer is that they'll stimulate, just sort of slowly. So if we try to activate, here's the control. If you activate with CD3 stimulation, here's what it should look like. So CD25, CD71, cell size are all measures of activation. So they pop right over, showing that they're activated. But our TILs, they kind of activate, but not great. And if we look at proliferation over here on the right, this is, sorry, here's your control. So we're just looking at proliferation. So you gather peaks with proliferation and in response to stimulation. So they stimulate like that. If we take blood from a patient with renal cell carcinoma, their T cells will stimulate just fine. You can see it looks just good. But we take them from the tumor and they just don't. Okay, so we've got a somewhat defective group. So we asked, why are these cells defective? And our first thought was really that it was the cancer cells who were hogging all of the glucose and that was making them unhappy. And so we actually started measuring how much glucose is in that space. So this is a measurement of glucose in the interstitial fluid space. And there was not a significant difference. There was more lactate. So maybe it's not a deficiency of glucose, but maybe it's a little bit too much of this toxin. That's possible, but it's not a huge difference. But interesting that they're not running out of glucose. We also checked other things, kind of if there's glucose around, is there GLUT1 so that they could take it up? Control is in gray and the tumor infiltrating lymphocytes are in the outline. There's plenty of GLUT1, okay. Hexokinase 2, that's the first rate limiting enzyme. That was there too. And phospho-S6, actually, if anything, was a little bit high. So, so they had what appeared to be the signals to take up the glucose. They had the glucose out there in the nutrient, but they weren't really using it. And so as we went kind of really diving deeper into what was wrong with these T cells, we found that their mitochondria were all really defective. So this is what the green here shows mitochondria. This is CD8s from a control. Mitochondria are normally nicely fused and large, robust looking mitochondria. On the right is a bunch of patient tumor infiltrating lymphocyte CD8 mitochondria, and you can see that they're very fragmented and spread all around the cytoplasm. We also showed this in a bunch of different ways, but this is now showing it by EM. So here's the control, nice normal looking mitochondria, but the mitochondria in these T cells are all really, really small. So we know that they're metabolically deficient in some way. These mitochondria are also hyperpolarized. So this is normal. This is the polarization state of the tumor infiltrating lymphocytes. They have higher levels of mitochondrial reactive oxygen species, but a little bit less of cytoplasmic ROS. So here's how much higher. So they're making a lot of ROS, so they're not neutralizing it. So that opened up some ways, like, can we rescue these cells by overcoming whatever one of their metabolic barriers there are? So our assumption was still that somehow the cancer cells were making them unhappy. We don't know how. And so we tried using MitoQ. That's a scavenger for reactive oxygen species. So it just sops up some of that ROS. And lo and behold, actually, they respond better. So this is one of the features that's holding them back. And so with MitoQ treatment and stimulation, now they'll activate a little bit more. So we still didn't understand why they had so much ROS. And so we thought it was possible that while there's glucose out here, but they don't seem to be actively taking up glucose, maybe we could just bypass that and give them pyruvate. This is not a great strategy for treating patients. This is really just a proof of principle cellular experiment, but we gave them pyruvate. And when you fed them pyruvate, you bypass all of that glycolysis step. And lo and behold, they also now stimulate. So you can see they stimulate much, much better when we feed them pyruvate. And if we blocked glutamine. So remember when early on I said T cells, if you block glutamine, will shift to preferentially taking up glucose. And so showing that here, if you blocked glutamine, then now they're going to be forced to choose glucose as their energy source, and now they'll activate as well. Actually, that's published now. Need to update that slide. So glucose is around, but the TILs, especially these CD8s, can't activate. Their mitochondria are abundant but are small and fragmented. That's really a downstream product of the fact that their metabolism is somehow uncoupled, and they can be partially recovered by treatment with pyruvate or glutamine inhibition. So the pieces are there for them to work. Their signal is just not quite right. And there is something about them living in this toxic environment that is making them essentially turn off their normal processes for activation. All right, now the home stretch is asking so all of this is like, we looked at a lot of tumors, or tumor cells, separate from anything else. That was a lot of studies where we take out T cells and we look at the T cells in isolation. So what if we look at the whole tumor? Okay. And the question was, who's really getting the glucose? Actually, at this point, we're still thinking that there's some competition going on, or there's something that's holding these T cells back from being able to activate. And is there competition for glucose? So if the T cells require glucose metabolism, is it available in all the tumors? Is the question. And we're going to look at T cells as well as myeloid cells. And I'm going to skip this slide. Well, I think I already showed you this. Some tumors can be very deprived of glucose regionally and very have very high levels of lactate regionally. So this is not a kidney tumor, but showing that there can be regional variation. This is just another experiment where we showed that interstitial concentrations of glucose and lactate, that there's plenty of it there, but it may be distributed unevenly in the tumor. Okay, so this experiment was really very simple. Was basically, we're just going to feed FDG to, not feed, inject FDG into a mouse who has a tumor, take out the tumor, and then see which cells took it up. Okay, so we implant a tumor into a mouse, and we do a PET scan, so you can see there it is. The tumor's taking up some FDG. This is not a kidney tumor, by the way, although we will show it later. This is a colorectal model, but this is in a mouse that has an intact immune system. This is a syngeneic model. We take out that tumor, separate using a magnet tumor cells, tumor infiltrating lymphocytes, myeloid cells, and then separately look at spleen cells, spleen T cells and spleen other cells, and count radioactivity. So let's put all these up here. So what we saw was that the FDG, here's how much was taken up in the spleen, whole tumor, isolated cancer cells was roughly at the same level as the whole tumor, and a lot more on a per cell basis showing up in immune cells. We tried another colorectal cancer model, got the same results, more going to the immune cells. This is Renka, a kidney cancer model, more going to the kidneys, and this is a breast cancer model, and we saw also here too. So in all of these models, when we inject with FDG, the glucose on a per cell basis is landing in the immune cells. A bunch of other models were all tested as well, all with similar results. Now, that's a really simplified way of looking at a tumor, like tumor cells, immune cells. There's a lot of other cells in here. MC38 is a colorectal cancer model. It's got, if you look at the spleen immune cells, spleen cells are mostly going to be B cells and some T cells and some other stuff. Here's what the immune cells of the tumor look like, right? It's a big mixture. All the red and pink are some kind of monocyte, myeloid, or macrophage. All the bluish ones are T cells. All the greenish ones are B cells. I guess yellow and orange are also in the myeloid family. So there's a bunch of different ways that we can classify these cells. So, and there's a lot of different kinds of cells in these tumors. This is in colon cancer, this is in Renka, and this is in a breast cancer model. But the other thing to look at here is there's an awful lot of macrophages, right? All of these are macrophages and monocytes. So all of these models have a lot of these cells that we don't think about a lot. I'm not going to show that. This is just to show that all of our assays had a lot of purity. So, okay. Oh, is that right? Is that what that showed? Oh, no. Sorry, I guess I took out the purity. So when we now look a little more carefully, separate out our subtypes of T cells, separate our subtypes of myeloid cells, we saw that all that activity is actually landing in a macrophage type component. F480 is one marker of macrophages and showing that that's where the glucose seems to be going. Okay, so the glucose could be going there, but it's actually translating to these cells being more metabolically active than their counterparts in the tumor. So here we're fractionating out these myeloid cells in red, T cells in blue, tumor cells in CD45 negative means tumor cells in black and the whole tumor in gray. And you can see that these cells that are taking more glucose are doing more oxygen consumption and as well more extracellular acidification. So they are more metabolically active. When we do gene expression profiling on these subsets of cells in the tumor, we find that the tumor cells have a much more of a gene expression signature indicative of metabolism of amino acids and nucleotides and fatty acids, and they all cluster separately. CD45 negative over here. In orange and pink are myeloid macrophage. We call them TAMs, tumor-associated macrophages, and they have a lot more carbohydrate metabolism signal. Okay, so that all actually made sense. So why on earth would they have more uptake of glucose? We started to look at some intrinsic signals so phospho-S6 is evidence of mTOR pathway activation, and we see very higher levels in these CD11b. That's another example of macrophages. And when we treat with rapamycin to inhibit mTOR, we can decrease the glucose uptake, most specifically of these macrophage subsets. So we think that they're programmed, that it's actually an mTOR-driven program that tells these cells to be glucose avid. And here I'm also showing hexokinase 2, sort of evidence of glycolysis is downregulated if we hit them with rapamycin. But the amino acid transporter CD98 was actually most high in our tumors, and that's independently mTOR-driven signal. So there's some mTOR signal also here in the tumor cells, but it's giving them more of an amino acid transporter phenotype. So if the tumor cells have more amino acid transporter, then what do you think they might be doing? Actually, I think that's the next slide. I'm going to skip. So we did a glutamine PET scan. So instead of a glucose PET scan, a glutamine PET scan, and the glutamine now goes to the cancer cells. So this is here in the MC38 and here in the RENCA model. So we have glutamine going to the cancer cells, glucose going to the myeloid macrophage component. Actually, the slide I skipped was we decided to look at lipids, and C16 BODIPI is a surrogate for lipids for uptake, and by far the preferred carbon substrate for these cancer cells appears to be lipid. So they like lipid, or love lipid, like glutamine, and let the glucose get eaten by the immune cells, particularly macrophages. So I think I might just skip this in the interest of time. So we also showed, as I had talked about before, in the T cells, when you block glutamine, you can increase glucose uptake. So now we asked, what if we do that in the whole tumor? So V9302 is an inhibitor of ASCT2, which is an amino acid transporter, so it blocks glutamine uptake. And so here, when we hit with V9302, we see glutamine go down. This is a control experiment. So glutamine uptake goes down when you block the transporter. And what do you think happens to the glucose uptake? Glucose uptake goes up in across the board. It goes up in the tumor cells, and it goes up in the macrophages, and it goes up in, this is a mixture of B and T cells here as well. So we really can influence these cells' ability to utilize nutrients and change the phenotype of the immune cells by altering the metabolic environment. That's the take home for today. So again, glutamine, cancer cells, glucose, macrophages, and the T cells like it too, but they have to be told to take it up. That glucose can promote, but glutamine can suppress effector Th1. Glutamine uptake in T cells can restrain glucose metabolism and prevent some of that exhaustion. And we can shift that by blocking glutamine and can allow cells to be more glucose avid. And then finally, what does the PET scan mean? The PET scan may sometimes really just reflect the inflammatory component and less so true tumor cell Warburg. So that's really basically the summary, and it's been a really fun ride. So this is my lab, and we work now a lot with Jeff's lab because it seems like the immune cells are playing a really important role, and I will stop there.
Thanks, Kim.
Yeah.
That was great. We just have a few minutes left for questions. I know you may have a few extra minutes. I have to jump off at 3.
No problem.
But yeah,
I'll stop share.
open it up to questions to the group,
There we go.
from the group.
So, the amazing talk, but just curious, the people several decades ago, maybe they're talking about FIH,
Yes. Yeah.
So do you have any comments on it? So it was such an amazing rise, and then nobody talking about that. But if you don't need FIH, that, that whole P300 complex with the HIF cannot do anything, right? I mean, so why you're not talking about that?
Yeah, well, I think it just gets really complicated. And so, so we have not done anything with FIH, although I've wondered about it a lot of different times. I think it's, HIF undergoes this prolyl hydroxylation, but then there are other modifications in there as well that may influence its stability, or which genes it actively transcribes, or what the rest of the complex is.
See, exactly my point. Doesn't matter stability. Final product is transcription, right?
That's right.
If you don't have that, P300 complex and that whole swang of transcription modulity, it will not work. So why you're not thinking that way, and what's the problem?
Yeah, well, so there were groups that were actively looking at that, trying to take HIF out by working through FIH, and I'm, I don't actually know why that kind of dissipated. I will tell you another story, just really, really briefly, that we're working on. So HIF itself, there are a lot of different post-translational modifications that can govern its stability. And so we've been looking at acetylation as well. And actually there's an acetyltransferase that when it's gone, then you actually can't stabilize any HIF2. And so in combination with a HIF2 inhibitor, you decrease the total level as well as the ability for it to complex with HIF beta. Yep. So I think that there are going to be multiple parallel ways to take out HIF, because there's this new drug belzutifan that is really exciting, but it's, we use it a lot in VHL, but it's a super slow drug. I mean, it's not melting tumors away. So if there's stuff that's, if there are components of the tumor that are really dependent on HIF, I think we need to be taking HIF out by multiple pathways. And FIH may be one way to get there.
Thank you so much.
Yeah.
Thanks.
Yeah, someday we'll be like the HER2 breast cancer people, hitting it from like all different ways.
Definitely. Anything else?